Hi, my name is Yash Soni, and today I'll be talking about our research on the sex-specific disruptions of LH pulsatility in mice with adult onset ER alpha deletion in kisspeptin neurons. The reproductive axis, as depicted here, is essential for the function of the reproductive system. While GnRH is considered the master regulator of this axis, hypothalamic neurons expressing the neuropeptide kisspeptin are the major upstream regulators of GnRH release. Gonadal-steroid hormones such as androgens, estrogen, and progesterone regulate this activity in this axis through feedback loops. Estradiol, a form of estrogen, binds to the estrogen receptor alpha expressed in kisspeptin cells, creating inhibitory and stimulatory effects depending on the specific hypothalamic nucleus. Disruptions in GnRH or LH pulsatile release may underlie reproductive infertility disorders such as FHA or PCOS. Previous studies in our lab use Cre-Lox-P genetic targeting to delete or knock out ER alpha, specifically in kisspeptin cells. These knockout mice, which we called Kirko mice, showed early puberty and increased levels of LH in females, resulting in impaired estrogen-negative feedback. Due to the developmental changes observed from this embryonic knockout, questions remained regarding the specific role of ER alpha in normal development, separate from its role in activational processes. So the main question of this study is to determine how estrogen signaling in adult mice regulates kisspeptin neuron function and how this affects LH pulsatile generation. Our main objective is to separate the organizational effects seen in the Kirko model from the activational effects of ER alpha signaling. To address this, we developed a novel tetracycline inducible Cree mouse model. This transgenic mouse model allows us to target genes in a temporal manner. With this mouse, which we call the iKirko mouse, we can knock out ER alpha and kisspeptin cells when mice are treated with tetracycline or its analogs. For our treatment protocol, iKirko mice were subcutaneously implanted with a doxycycline pellet, inducing Cree expression in kisspeptin neurons. To confirm KISS neurons have no expression of ER alpha, we use fluorescent immunohistochemistry in the hypothalamus. White arrows show the lack of ER alpha in KISS cells. Our preliminary findings show that the loss of ER alpha in KISS cells results in an increase in LH pulse release and frequency in females. This can be seen from the increase in mean LH and pulse frequency on the right, and as depicted on the left by representative LH pulse profiles of control and dox-treated mice. In contrast, males that lack ER alpha and KISS cells also showed an increase in LH pulse frequency, but they also saw a change in amplitude. This is seen in the representative LH pulse profiles. To conclude, our results suggest that disrupting estrogen negative feedback in adult postpubertal mice leads to sex-specific changes in LH pulsatile release, which supports the importance of AR alpha signaling not only in organizational processes but also activational ones. Thank you.